Hey, this is Russ Reed. I'm the Executive Director of the National Center for the Biotechnology Workforce here at Forsyth Technical Community College in Winston-Salem, North Carolina. And we're delighted. We have this series we call SciTech. We always love presenting it to you. We have another grant we call Beta Skills, which stands for Biomedical Emerging Technology Applications. And we did a professional development this summer. And one of our speakers, Dr. Todd Smith of Digital World Biology, based in Seattle, where Dr. Smith gives an overview of the supply chain. And the supply chain was particularly critical for the development, the quick development, the rapid development of COVID vaccines. I want to thank our sponsors, our many sponsors, and their logos will be at the end of each of these lectures. I hope everybody enjoys them.
Well, good day, everybody. I'm Russ Reed, and I'm the principal investigator for Beta Skills, which is Biomedical Emerging Technology Applications. And I'm delighted again to be with Dr. Todd Smith from Digital World Biology. And Todd, how are you today?
I'm good.
I'm delighted that you're going to help us today to define for the Cyberbio participants, a little bit about Cyberbio from the supply perspective. So why don't you take it away?
Okay. That sounds great, Russ, and I will share my screen. So what I want to do is just cover a few things. We're gonna work our way to a figure that I developed that talks about the vaccine supply chain. But first, since one of the things we do, with you is we create content to discuss the various topics in that are relevant to Beta Skills. And so one of those is cybersecurity. And we've presented biotech careers and other formats for people, but it's a good opportunity just to do a little bit of a review of navigation. So what I'm doing is going to blogs, and you can see one of the most recent ones by Sandra Porter is What is Cyber Biosecurity? And she reviews a paper that she saw that talks a lot about various issues in biotechnology that relate to cybersecurity. And what motivated this was, the Colonial Pipeline hack. It was a good opportunity to have everybody think a little bit more about cybersecurity in general, and cyber biosecurity specifically. So, in this blog, and I'm not going to go through the whole blog, but just to cover a couple of the key points in it, it's talking about potential for risk. So when people are in biotech companies, they're dealing with a lot of computer systems, and as people will learn in other discussions in cyber biosecurity, as medical devices, a lot of instrumentation are kind of gateways, holes for hackers, because some of them use old operating systems and things like that. So this is just a brief story that people can review on their own. If I go back to the page on cyber biosecurity, we have what's so hard about making a few billion vaccine doses. And this is what I want to spend a few minutes on. So what was the motivation behind this blog was, in the January timeframe, this fellow at Zymergen in California, one of the synthetic biology companies that just went public, wrote an excellent article about exploring the supply chain of the Pfizer, BioNTech, and Moderna COVID-19 vaccines. And he covered it in just tremendous detail. I recommend that if people are interested, they go and read that on their own. But one of the things that was missing is what we call the central figure, right? And so I read his blog, and I went through and I made notes. And from that, what I developed was a map about the vaccines and how they're made, and some of the points of information systems. So when I think cyber biosecurity and I think of biotech, that leads me to computer systems. And many have heard me talk about this and say this before, but the reality is students that are coming out of programs, they need to be much more aware of computer systems and working with computers in terms of LIMS systems, analytic systems, instrumentation systems, because after you've done some of the hands-on bench work, you're going to go into an environment and you're going to immediately start to face computer systems. So when we think about the Moderna vaccine and the Pfizer vaccine, the supply chain is actually very fascinating because these vaccines and the components, so the first component in both vaccines, as we know, they're RNA vaccines, but we make RNA from DNA, right? So we use transcription, and we use in vitro out of the body transcription to make the RNA from the DNA. So the first step is you're going to make DNA, and that begins with the synthetic DNA. And this is done by people who are genomics technicians, molecular biology technicians, and you can click on each one of these links in this figure. And what it's going to do is take you to the respective place in biotech careers. So, if we want to know more about a molecular biology technician, we just click that and we come to the molecular biology technician. A genomics, same kind of thing. And so it begins with synthetic DNA. So simply, we can just, from the COVID sequence, from the SARS-CoV-2 sequence, I should say, we just type that DNA for the spike protein region that we want to make a vaccine to into the computer, right? And then we send this off to a company. It could be Twist. There's a variety of companies that make synthetic DNA. We get our DNA, and then we're going to put it into a plasmid. Because what we need to do is we need to make this DNA at scale. And synthetic DNA is not too expensive as long as you buy just a little bit of it. But as we know from our molecular biology, we can clone this into a plasmid. We can put that plasmid into bacteria. So these are all the basics that are covered in biotech molecular biology. And then that bacteria is grown in fermentation processes. So this is going to be your biomanufacturing. This is going to be the upstream biomanufacturing that we learn about at places like VTech in North Carolina and others. It's still a major activity, lots of fermentation. Facilities technicians are going to be the people who are kind of working in an environment that oversees the process. And then we grow up the bacteria, and then we purify the DNA. So we have upstream and downstream biomanufacturing. And so, again, different kinds of jobs. What's interesting here, though, is what we need to make these RNA vaccines is we need gram quantities of DNA. And the other interesting thing is I didn't talk about it here, but, one of the, and is it science fiction? Is it reality? This is a place for hacking, right? So you could change, if you could get into people's synthesis machines, you could change the DNA sequences that are being produced. The reason why I asked the question science fiction or reality is for a hacker, somebody who's going to break into a computer system, there has to be generally an economic motivation. So, that's not necessarily the case here, but it can also be data entry. There's a lot of errors that can happen. So there are security elements. So now we make gram quantities of DNA. Now what we're going to do is we're going to make mRNA from those. And so we need to do very large scale mRNA preparation. And there's some videos that we can attach in the canvas for Pfizer, what they're doing and how they scaled this process up. Because in molecular biology, we're used to doing these things at small scale, but now we need to make a few billion doses, so we need a lot of mRNA. And in this figure, what I also have is in red are places where there are rate limiting steps, okay? And that is the capping enzyme. To make your RNA, you have to cap it. And it turns out, in Jonas's blog, and this, may be changing now, but there's pretty much one company that makes this capping enzyme. Even though you could find other companies that distribute the capping enzyme, when you start to navigate the supply chain, what you find is sometimes there's a single source. And single source is a vulnerability in any manufacturing process. And this is something that supply chain logistics managers and people that work on these processes, they look for these kinds of issues because those create risk for the company. So capping enzyme. Lots of people make polymerase, you need buffers, you need nucleotides, and this fancy prep, and then what it's going to produce is mRNA. And so the endpoints are in blue, and so the mRNA is now going to go into this special place called the nanoparticle mixing chamber, right? So we don't vaccinate people with raw RNA, we package it in nanoparticles, which are really a particle that's comprised of a complex lipid that comes from several different sources. And here's some companies that make those particular lipid sources. They're combined, and they're combined into these nanoparticles. As Jonas said in his blog, you just don't take mRNA and lipids and put them in a Vitamix and get out nanoparticles. No, you need special microfluidic technology, and you need people that know how to operate it. And it turns out this is still a limited area. So it's an impact risk. There aren't a lot of people that know how to operate these devices. Operating them at scale is another challenge. And there isn't a lot of this equipment, right? So this is another place of impact risk. But as we know, and many of us have had the Pfizer and Moderna vaccine, so we are getting through the process. We get the nanoparticles, and the next step is they need to be packaged into vials, and the vials need to be labeled. And one of the really interesting problems here in terms of impact risk is boron. The available boron in the world is also a limiting factor. And why do you need boron in these glasses? Well, the glass vials need the boron because when you mix boron with silica and make a glass, it is able to expand more, right? And so when you think about it, these vials are going to be at room temperature. They're going to be at -80. So they go from a normal temperature to extreme cold, and we really don't want them cracking because that would be a negative, okay? And so the vials, we get the nanoparticles, they get packaged here. The vials need to have a label, and I'm going to come through the computers and IT after we walk through this. And then we put them into cartons and trays. So Moderna has a different configuration than Pfizer. And then these need to go into the cold cases, okay? And the cold cases have GPS devices and things. We need to track where these things are going in the world so that we know they're getting to where they need to go. And then dry ice. Because they need to be cold, we need a lot of dry ice. And dry ice, it turns out, because a lot of factories that make dry ice are next to petroleum plants because they put off CO2, and dry ice is just frozen CO2. We get dry ice from petroleum. Well, during COVID, petroleum went down, so dry ice went down, and I forget which. I think Pfizer went to making their own dry ice. I mean, so it kind of speaks to the issue that dry ice is a rate limiting step in this process, and really what drives that is petroleum. And then these cold cases go off to kitting. This is the last step, and it's in distribution. And Jonas spends a few paragraphs on this, and it's really great to understand. But he talks about the whole Operation Warp Speed. Pfizer chose to handle their own process. They didn't join Operation Warp Speed where Moderna did, and because Moderna joined, they go through a different distribution process. But basically the kitting, which is a biotech career, people putting together components to create a proper kit that can be used in the field with all the components you need, involves the vials, involves the syringes, gloves, masks, and then this goes into vaccine kits, which then get distributed to pharmacies and football stadiums and all the places where people are being vaccinated. And having enough doctors and nurses to do that is one of the challenges in making sure that we can get a few billion people vaccinated. And then you have a vaccinated person. And then the last part of cybersecurity is the vaccine card, right? And so through this process, what we've seen is we have companies working on COVID-19. We have companies that work on LIMS and automation systems, contract manufacturing. And speaking of contract manufacturing, let me back up. So I've got a pink box and blue boxes. So Moderna contracts this whole process out to Lonza. And Lonza does in the US, they work in Portsmouth, New Hampshire, and then in Visp, Switzerland for Europe. Pfizer, they don't contract, but they do the process in 3 different places. They do it in St. Louis, they do it in Andover, Massachusetts, or in Germany because they're a partner with BioNTech in Rensselaer, Germany, or in Kalamazoo, Michigan for the US is where they do the lipid nanoparticle packaging and other worldwide locations. So through this, materials have to move from St. Louis to Andover to Germany to Kalamazoo, and then out to the world where people get vaccinated. the whole process, when you're started, you're going to be making batch records that need to track these different steps and movement of materials, and there needs to be lot numbers. So when you get your vaccine card, they're going to write a number on that because the label has a number, and that tells you which lot of vaccine you received, which comes from the lot numbers and all of these different batch records. So controlling this then is a quality management system that's composed of LIMS systems and other kinds of computer systems with a lot of barcodes and tracking going on. And then these QR codes go on to the labels. So that was just the LIMS automation systems. And then, in biotech careers, you can look at vaccine companies, DNA, RNA synthesis companies. So, altogether there's, technology from thousands of companies that go into these processes. And it's also really important, I think, to illustrate that a biotech product is not a single company thing, right? When somebody makes a product, it's coming from a lot of different places. You have reagents, you have technology, you have all of these different components. And so there's a real ecosystem going on. So, so going back to the figure then, if we wanted to know the computers and systems, we kind of look at, we can click these buttons on the bottom, and they're going to highlight these areas in the figure. So we can turn on the yellow, and this is going to tell us where all the different computer and systems are used. If I turn that off and I click cybersecurity, I get the same thing, right? And that's because cybersecurity, anytime you have computer systems, you have security issues. And one of the things when we think about batch records and electronic signatures and these kinds of things in a quality management system is the technicians, the people operating these systems. There's a lot of validation and a lot of the systems control about who can do what when within the system. And so all of that goes into your GAMP, your good automated manufacturing processes. And the companies that deliver these systems, they have to deliver them with documentation and the ability for their partners to validate those systems. So you have validation specialists. They're going to be involved with cyber biosecurity because part of validation is making sure you can't inappropriately access a system, that the system has some security against invasions. The company should have a firewall. The system should be behind that firewall. But even with that, the software running those systems, let's say it's a web-based data management or LIMS system, LIMS being laboratory information management system, those are URL accessible. So the software itself has to have protections against unwanted access to that software so that people can't, put in inappropriate records, these kinds of things. So you have validation specialists, the QA assurance, QA document coordinator. They're going to be interfacing with a lot of different software systems as they go through this process. The labels, this was called out as a cybersecurity because these QR codes, if they get hacked and people put the wrong kind of label on vials, that can be a mistracking. It's also, we've had stories of, inappropriate vaccines. So fake vaccines being put out into the market. So the labels are, it's just a piece of paper, but it contains this data that's in this QR code, and it's that data that can be substituted. The cold cases, why are these here? Well, the cold cases have little GPS devices, and so, GPS, it's, there's a book on, software systems and usability design, and it goes through some riddles at the beginning. And the riddles are always, what do you get when you cross a computer with a car? You get a computer. What do you get when you cross a camera with a computer? You get a computer. So it always ends in a computer because it's a computer, right? And so the cold cases are little computers with those GPS devices. And then finally, the vaccine card, this has personally identifiable material on it. And there's been a lot of stories about, don't put your vaccine card on Facebook. Do tell everyone you got vaccinated. That's important to do, but don't share your card on Facebook. So that's why, when I turn this off and I go computers and systems, turn that off, do cybersecurity, you end up with the same things. It's that, to extend that riddle from what do you get when you cross a computer with a device, you get a computer. What do you get when you have a computer system? Well, you have a cybersecurity issue, right? So you need to you need to know that any computer system is vulnerable. The other is, we call out just the general components. So again, just to reemphasize what's going on here, to make one of these vaccines, you need a lot of different materials, right? So synthetic DNA, we need plasmids, we need bacteria, we need polymerase, we need a ton of materials as you go through this process. Each one of these, a supply chain logistics manager or technician needs to be able to identify the sources of the materials, the availability of those materials, and be able to have more than one vendor to have the most robust system as possible. There's several kinds of processes in this. We have fermentation and purification, mRNA preparation. I call this mixing, but remember, it's very, very complicated. Kitting, is just some of the processes that are involved. And then there's the endpoints, and the endpoints are, each station creates a product, right? So from synthetic DNA, we needed to make gram quantities of DNA. Those gram quantities are used to create RNA so that we have a lot of RNA. We need to make a lot of nanoparticles. And so these endpoints are the things that flow through the system to finally get to the vaccine kits to get out to people. And then through that, we had several kinds of what we call impact and risk. So in any supply chain logistics, you need to think about where are my risks, because those are the things that, if you can identify those well, those will be the pieces that from a product management and logistics perspective, you're going to focus on and monitor the most close, because you need to be ahead of the game. If you're getting any sense that these guys are getting low, you either need to get in there and buy a whole bunch or figure out another plan. Otherwise, you could find out that we're just not going to be able to make mRNA today, and that's just not good for getting people vaccinated. So that's the supply chain. And as I mentioned, you can click on these links, and it's going to take you to the appropriate pages in biotech careers. But anyways, in biotech careers, we have now 284 employers that work on vaccines, and it's not surprising that 166 of them were working on COVID-19 vaccines, because this has been the problem of the day, as it were, for the past year. And it's really, when you think about the production of a COVID-19 vaccine, we went from none to several vaccines. There's 4 out on the market. 3 are particularly good for all the variants in just over a year. So that's an amazing accomplishment. For perspective, mumps was the previous record, and that took 4 years. So that's the vaccine supply chain in biotech careers.
Really, really good overview. Now, I just had a question for you.
Yeah.
When you took this person's article, did you then map it out? Is that what you did, or was that completely something
Yes.
that you? Wow.
Yes, that is what I did, and then.
Great.
Yeah, and then I followed up with him. Because we when we published it, I reached out and sent him a link. We connected on LinkedIn, and he loved it. And actually, he wants to help us with some of the automation technical skills that we talk about in biotech careers. So he's on the software side at Zymergen, but they work on automation systems, and so he was very enthused about the figure and the site, and then Sandy tweeted it out to one of her science writing colleagues, and he tweeted it, and we've had thousands of views of that article.
That's great. It's interesting because a previous boss of mine was actually on warp speed and helping out with distribution, and so he very early on into the process, I told him that I was in the clinical trial for the vaccine and with Moderna, and he said to me, cold storage, cold storage issue, and brought it right to my attention. And so I think that what what is going to be really cool for our folks when they see this and they study it, you know, looking at all the possibilities that you've outlined where there could be an intervention and not so nice intervention, you know, really does freak you out to think that, oh my gosh, they did it. You know, they were able to protect the supply chain, which is pretty cool.
Well, it is. And when you think about making those vaccines, I mean, in such a, you know, it's not like we just started working on mRNA vaccines, but in any vaccine, you know, the supply chain logistics are really complicated in pretty much any biotech product, you know. I think sometimes this is an underappreciated aspect, but as we've been seeing on Beta Skills, and we have, you know, people in from Cook and some of the other companies, when they talk about the jobs they have, this is always one of the top ones that they need to fill.
Yeah, exactly. Well,
Yeah.
Todd, I want to thank you for coming to our Cyberbio security workshop, and thanks very much for doing this. And I know that if anybody needs further information, they can always reach out to you. And a good place to go is either the Innovate Bio website or go to biotech-careers.org.
Yes. All right. Thank you.